Good morning, guys. So this is our second to last week of material for biology, with Christmas break and then having to get a test in before our final exam at the end. We don't really have a lot left. So we have this week where we're going to learn about chromosomes and meiosis, and then next week we're going to learn a little bit more about genetic mutations and Mendel, and then the week after that we have a test and then our final exam, and then we're done. And next week, I mean we're going to take the 2 weeks for Christmas break, and then that's the first stuff we're going to come back with after Christmas break. So that means that over this week and the 2 weeks for next week, you guys have a lot of time to catch up on any assignments that you have missed, any quizzes, any discussion questions, any assignments. Make sure you turn them in. I'm going to be accepting work up until the very end, but it's just a lot of pressure for you guys to try to turn everything in the last minute. So I just want to make sure that you're out there and trying to get information turned in. Also, there are makeups. There are makeup tests and there are makeup quizzes for all of the quizzes that we have taken so far, and for the tests. So please make sure that you are doing those. And if you do the makeup test to get a higher grade, please make sure you email me so that I know to go check to see that you've done them. It's really for you guys to get you the highest grade possible. That's my goal here, is that you guys are being successful in this course, and I just want to provide that to you. So please make sure you're doing those and then email me once you've completed them. If you have any questions about anything, please reach out. The rest of this week I'll be working. Over the break, probably not so much. But if you reach out and I just by chance check my email that day, maybe I'll send you an email back. And then when we get back from break, yes, please reach out so that we can make sure that you're set and ready to go for those final 3 weeks and getting everything done and turned in. All right, so like I said at the start of this, we are going to be working on chromosomes and meiosis. So let me pull up that slideshow and get started for you. All right. So last week, oh, not what I wanted. Last week, we learned about mitosis, which is very similar to meiosis. It's just a little bit different. We're still going to follow that PMAT that we talked about last week, but meiosis is more dealing with gametes, is more dealing with sperm cells, egg cells. So we're just going to really get into that. There's also 2 processes for meiosis. So let's jump right in. So when we're talking about genetic variation, it's truly and honestly, it is essential for any species to evolve. Without genetic variation, it is most likely that genetic organisms will die, because only the fittest survive. That's Darwinism. That's just kind of basic life. You see those comics all the time about like the giraffes with the short necks, and then they end up dying off, or the giraffes, then the next giraffe got a longer neck so that he could reach the leaves higher on the trees. Doesn't exactly work like that. But yes, those that can reach food, that are faster to get away from predators, that can blend in with their habitat, those are the ones that are going to survive. And that's what genetic variation provides for us. Sexual reproduction, which we covered back in Unit 2, helps us to ensure that genetic variation occurs. The crossing over and independent assortment create genetic variation through random fertilization. So making sure that we're always getting random DNA. So what is a chromosome? A chromosome is just a condensed or a smaller form of DNA. Okay, and they look like this. So these little X's kind of things, and they contain our DNA. What are the different parts of the chromosome? Up here, you have the sister chromatids, you have the centromere in the center, and then you have the chromatid, that squiggly stuff right there, down at the bottom. So the chromatin is just loose strands of DNA and a little bit of protein. The sister chromatids are exact identical copies of the chromosome. And then lastly, the centromere is just the point in the middle where the two chromatids are joined together. So karyotype looks like this. It has all of the chromosomes. The very last one down here is our 23rd chromosome. It's this here, it's this XY, and that distinguishes what our gender is. So a somatic cell is any cell that is not a sex cell. So all of these other ones are somatic cells, and they're present. And 46 chromosomes are present in all somatic cells. In egg and sperm cells, so this X and this Y, there's only 23 chromosomes present in there. Haploid cells, or egg and sperm cells, contain 23 chromosomes. Diploid cells, these ones over here, contain 46. So you have these diploid cells over here. Together, the sex is a diploid cell. And then you have a haploid over here, okay? So these are all halves. And so then you have just the Y over here. All right, so now let's take a look at these pictures here. We didn't really talk about it, but so X is a distinguishing factor. If you have two X's, it's a distinguishing factor of a girl, and if you have an X and a Y, it's a distinguishing factor of a boy. So which of these two pictures would be a girl and which one would be a boy? Yes, this one over here, where you have the X and the Y, would be a boy. And then this one where both of the chromosomes are lined up on the X, that one is a girl. That is a big reason for a lot of genetic variation. So for color blindness example, it's very, very rare to find a female who is colorblind because it is typically carried on the Y chromosome, color blindness, and girls don't carry that. So it's very, very rare to find women who are colorblind. Sometimes, some of the color blindness can be carried on other genes, and so that's where you will find a woman who is colorblind, but for the most part, it's carried on this Y gene over here, and that's why you typically only find males who are colorblind. This is a video for you guys that you guys can watch later. Okay, so now let's jump into the process of meiosis. We already talked about mitosis, and that kind of gives us the basic steps of it. So we have prophase, we have metaphase, we have anaphase, we have telophase, and then cytokinesis, splitting of the two cells, right? So in meiosis, we have all of those same steps. So PMAT again, but in meiosis, we call it PMAT 1, and then we do it one more time, and we call it PMAT 2, okay? So meiosis is plain and simple the reason why you look a little different from your siblings. You have slightly different genetic information, unless you're identical twins, of course. So your parents are, one's going to give a sperm, the other one's going to give an egg cell, and they're going to combine together. And every time the genetic information is going to be a little bit different, and that's why you look different, but also similar to your siblings. So meiosis is the process where the sperm and the egg cells make gametes. So they make the baby cell, okay? So you have the egg cell, then you have the sperm cell, they combine, and then you end up with a bunch of baby cells, okay? So these are what they're trying to distinguish here. These are the chromosomes combining together to make the baby cells. Sperm and egg cells do not have 26 chromosomes. They are always haploid cells, so they always only have 23 chromosomes. Okay? Again, haploid cells, sperm cell, 23, egg cell, 23. Combine them together, and that's how we get to 46 chromosomes in all of your cells. All right, so meiosis means the reduction of division. So in meiosis, we are reducing the number of chromosomes. There's still interphase when we're talking about meiosis, just like in mitosis. So in interphase, it happens before the process of meiosis. We still need the cells to grow, and we need them to be able to copy their DNA. Otherwise, there wouldn't be any more cells. And so then they still have, as those cells are growing, they'll still have 46 chromosomes, but then they'll have 92 chromatids. In meiosis, you just divide them twice. And we put numbers at the end of the phases to indicate if you're in the first stage of it, so the first PMAT 1 or PMAT 2, second division. So in meiosis 1, what you have here is you have the state of interphase, then you have prophase 1, where this is where the genes are crossing over, okay, creating that mixing process. This is where you start to look a little bit different from your siblings. And then you have metaphase 1, and then again you have anaphase 1, where you're going to have some independent assortment, so they're not going to divide exactly, evenly. Again, another reason why you look a little bit different from your siblings. Then you have the segregation, and so that's where the homologous pairs are separated by reducing the number of chromosomes in half. So and then at telophase, you end up with 2 cells. So then you jump down into meiosis 2, and you have prophase 2, metaphase, again, they line up in the middle, and then in anaphase, they start to separate and split apart, and then you have telophase 2 down here. So this is where the sister chromatids are finally separated, and it produces 4 haploid gametes, okay? All right, so let's talk about prophase 1. So what happens in prophase 1, everything condenses, thickening, same as prophase in mitosis. They line up with the homologous pairs, the same types of genes in the same locations. This is where crossing over happens. Again, that's the transfer of genetic information from one chromosome to another. This is a big creator of the variety in siblings. So here's a little better example to explain crossing over. So you start out, and these look totally different, right? So you got one from your mom, one from your dad, or one from your mom, one from your dad. And then when they're crossing over a little bit, they mix, and now, oh, I got a little bit here, and I got a little bit here. So now this is why. Now this one's not totally blue, and this one's not totally red. Okay, it's got a little bit of blue and a little bit of red. So then in metaphase 1, the chromosomes line up in the pairs in the middle of the cell. Again, same thing as mitosis. So here's what metaphase and mitosis looks like. In metaphase 1 of meiosis, it looks a little different because you're just going to split the 2 sets of chromosomes in half, not split them, not split the single chromosomes in half. So you're just getting the cells to the separate sides. And then in anaphase 1, the chromosomes are being pulled away from each other. So if we think about in anaphase in mitosis, it looks like this because half of each is going to each side. But in anaphase 1 in meiosis, say you have this, it's like the 2 are going to both sides. And so that's why you see the 4 legs on each one, because it's actually whole chromosomes going to each side. And lastly, in telophase 1, 2 newly formed cells, and they have their own nuclei, are formed in telophase 1. So then next we jump into PMAT 2, or the second half of meiosis, meiosis 2. And so in meiosis 2, we're going to remember that this time when we're actually dividing the amount of DNA in half. Instead of having pairs lined up like we had the last time, the individual chromosomes are going to line up, and then they're going to split down the middle. And once they split, they're going to become haploid cells. So half the number of chromosomes, haploid cells in humans are going to have 23 as opposed to our full 46. Okay. So here you go. Here's paternal DNA, chromosome from your dad. Here's some from your mom, and the DNA replicates. And these ones seem to look like they stayed. They didn't have any crossing over at this time. Okay. So then this one didn't cross over. And so over here, what you have is cells at the bottom that are going to look exactly like what dad gave. Okay. And over here, over here, you'll have cells that look exactly like what mom gave. Sometimes that happens, but for the most part, we do have some crossing over here. So dad gave you a cell, mom gave you a cell. They come down here, and they're really friendly with each other, and they kind of switch some DNA up. So then you come down here, and you've got this cell that's got It's got a little bit of, this one looks exactly like dad, but this one's like, oh, I kind of look like dad, and a little bit of purple like mom. And then over here, you've got this one that says, oh, I look a little bit like dad, or a little bit like mom, and a little bit like dad. And then this one over here is exactly like mom. So that's why you have some traits that, you know, are very similar to features that of your dad, and then some traits that are very similar to features of your mom, because of this crossing over process. And then some features, you know, you kind of look at you and you're like, I have no idea why I look like that or have that. And that's, you know, another part of this, like a mix between mom and dad, where you can't distinguishly see who it comes from. Meiosis creates a great variety. So the exchange of chromosomes during segments happens in the chromosomes. During phase 1 of meiosis, independent assortment happens with sperm and egg cells contain half the genetic information. These genes then get sorted independently. So your mom gave you a gene for eye color, and so did your dad. So you have 2 genes for eye color, but only one of them gets passed on to your children. Okay? And you only pass on one to your children. And that's everything when it comes to meiosis and mitosis. It's a very brief snapshot, but it's really important to know why crossing over happens, why we have that genetic information. I'm going to jump in here and see if there's anything in our classroom that we need to cover. Assignments and information. Okay, don't forget to do the discussion question and then the quiz this week. Okay, and like I said, and then the other thing you guys should know is that I set these dates. You can do them whenever. It would be great if you got them done before Christmas break, but if you don't and you want to do them over Christmas break, I totally understand, or the holiday break, I'm sorry. I totally understand. I didn't set these due until January 4th. So that gives you a lot of time. That gives you a whole 3 weeks to get them done. Okay. All right. If you guys have any questions, please reach out, email, chat, whatever works for you. And I will see you after break.